Okay, guys, going to start on chapter 6, acellular pathogens. When we say acellular pathogens, we're pretty much well referring to viruses. Viruses are kind of unique. These guys are super small, even smaller than the smallest of the bacteria. We can't see these with our light microscopes. They're that small. These guys are not a cell. These guys are a piece of DNA or a piece of RNA or two pieces of DNA and two pieces of RNA. That's it. Now these guys usually they're packaged inside some sort of protein capsule, and that's the virus. These guys infect us, they infect other animals, they infect plants, and they even infect bacteria. Most of the diseases that we deal with today are virus. And of course, we know that with coronavirus having just, been a major problem. These guys do not do any biochemistry because all they are is DNA and RNA. They can't grow because they're not alive. They don't respond to the environment. They cannot reproduce all by themselves. They have to actually infect the cell. Once they get in there, they will take over the ribosomes and all the other cellular machinery and use that to make copies of themselves. Uh, no plasma membrane, no organelles. Now, these guys exist, of course, inside the cell, intracellular, and then the out extracellular, which for many of these guys is going to include being inside that protein capsule. Characteristics. Well, as far as the extracellular state, this is important, guys. Virion. A virion is what we call a virus when it's still outside the cell. Why am I doing this? Because there is such a thing called a viroid, which is a virus which has no capsule. We talked about that before. No capsule is just a naked piece of RNA. This is a plant virus. So don't confuse virion with viroid, okay? So they're in that protein coat, which is called a capsid, surrounding the nucleic acids. When you call them all together, nucleocapsid. Some of them even have a phospholipid. So there's the nucleic acid. There is the protein coat. Some of these guys, when they leave the previous cell, actually take a bubble of plasma membrane with them. And that is really like the wolf in the sheep's clothing. And that's the point. It helps them to avoid our immune system. So that's that phospholipid envelope. Outermost layers, a lot of these guys, and you probably have seen those little spikes. Those are proteins, and for some of these guys, these are the locks that open up our cell. HIV has these proteins, and they are used to get the T helper cells of the immune system to actually bring them in and help them to infect them. It's like a key to a lock. Protection and the recognition sites. The intracellular state, this guy generally does not come into the inside of the cell. Sometimes it does, but if it does, it's really kind of short-lived. Most of the time, these guys inject themselves in or endocytosed, but the capsid itself is really of no use. It's just a means of transport for the virus. And Well, that goes without saying because that's what we said. The capsid shapes are kind of wild. You've got this one, which is a spiral shape, and they call it helical. We've got this one, which is complex, which doesn't follow. But this one, the icosahedral, and again, guys, please remember, I'm not an artist by anybody's standard. Anyway, that's it, it's icosahedral. Let's just leave it at that. I can't draw that. So the capsid protects them, and the capsid has those spikes on it that helps them to get in there. These guys are made up of subunits called capsomeres, and a capsomere can be either a single or multiple types of proteins. Genetic material, we already said they can have double-stranded DNA, single-stranded, double-stranded RNA, which only exists in these viruses, and single-stranded RNA. Linear, segmented, single, circular, and generally they're much smaller than even a bacterial's chromosome, its genome. Host of the viruses. A lot of these guys will only affect one type of cell. You have the hepatitis viruses. There's hepatitis A, B, C, D, and E. These guys have an affinity for liver cells. HIV has an affinity for the T helper cells. There are certain viruses that are very specific, and there are others that are even some that there are a lot of viruses you cannot give your dog, and your dog cannot give you because they're so specific for that type of cell. And on the other hand, you've got the flu, which starts out with birds and can pass to animals and finally come to us. Those are going to be the general list as opposed to these really highly specific ones. A bacteriophage is the virus that attacks and infects a bacteria. Plant viruses, a lot of these hit things, crops we grow for food. Fungal viruses are not understood that well, so we're just going to pass over that one. The envelope is when they do exocytosis, the capsid, then they'll take a little bubble with them, and that will help them to hide out. Some of them will still have the spikes, but some of them won't. So there's an envelope. That's what we call an envelope virus. There is the capsid. The RNA, DNA is going to be in there someplace. This is actually plasma, a phospholipid bilayer, and this was taken from the cell that it was infecting as it left. And there's a transmission electron microscope of it, one. As far as how do they classify viruses, there's a group that does the taxonomy. They classify them by the type of nucleic acid they have, whether they're enveloped, and if they're not enveloped, they're considered to be naked. A naked virus doesn't mean it is not like a viroid with a naked piece of RNA. A naked virus is simply one that has a capsid and no envelope. By their shape, their size, relationships, there are no kingdom, divisions, or classes. Again, these guys are not living creatures, so they kind of do this. So now we're going to talk about the bacteriophage. This is a virus that attacks and destroys bacterial cells. Usually they're just going to fill the inside of the cell up with copies until the cell bursts, and that's the lysis. This is something you definitely need to know. The 5 stages of lytic replication. The first thing the guy has to do is attach, and I want you to notice that this guy has got these spider-like legs. If you guys were a little older, I'd say it looked like a lunar module, but you wouldn't know what I'm talking about. And I'm just drawing the capsid like that. And inside is there. Now, these guys, these legs land on the surface of the bacteria, and then this guy, he can actually move those legs so that the capsid moves up and down, and he basically moves this down, and this center thing is going to puncture the plasma membrane and the cell wall of the bacteria, and that's how the viral DNA gets in there, or RNA as the case may be. So obviously the first thing he's got to do is attach to the thing. The second thing he's got to do is have that structure kind of sit down so that that central hypodermic-like needle thing can penetrate, and then when that happens, the viral DNA enters there. Next is going to be our synthesis phase, where what's basically going to happen is in some cases the virus is going to destroy the bacteria's DNA, and then from that point on the viral DNA will be running everything. But they're going to make all the pieces. They're going to duplicate the RNA or the DNA. They're going to use the protein ribosomes to make the capsomere subunits. These guys are then going to be put together to form complete virus particles in the maturation phase, and then eventually they're just going to lyse. And so that's what we've got down here, guys. Lysogeny, this is a modification of that bacteriophage. In this situation, what happens is the virus attaches, he injects his nucleic acid, and instead of destroying the DNA of the bacteria, he actually cuts it and inserts his right there. So the cell is going to keep dividing because that's what bacteria do, and every time they divide, they actually make a copy of his DNA. Now, this is kind of like, why doesn't he just kill it? Because he's like a cow. I'm going to get all the milk I can, and once the cow can no longer give milk, then I'm going to have a hamburger. Does that make sense, guys? So once this thing is unable to divide, has some sort of stressful condition, then the viral DNA is going to cut itself out, and then it's going through the normal lysis cycle. So lysogenic infection is only, I'm not going to kill you now, but I will kill you later when you're old and weak and can't be, you're of no use to me anymore. So that's the way this guy does. These are also called temperate phages because they don't kill them immediately. Prophage is what you call this piece right there that's inserted. It's the inactive. It's just laying low and letting the bacteria reproduce it with each passing of the cell division. Lysogenic conversion results when these phages, sometimes we talked about that piece of DNA being inserted in there, but we didn't talk about what could it do to change this. Actually, those genes, some of those will actually change the characteristics of the bacteria. Many of these will change these bacteria into a pathogen. Corynebacterium diphtheriae is pretty nondescript and pretty mild, but when it gets infected with a lysogenic bacteriophage, it actually gets the ability to make the diphtheria toxin, which is what gives folks diphtheria. So a lot of bacteria become disease causers, pathogens, after they get infected by a lysogenic bacteriophage. Transduction. This is what happens. Here's my bacteria. There's the viruses thing. Now, when this in the prophage, sometimes these guys are careless. So instead of cutting their DNA out, they'll actually get part of theirs, and they'll actually take part of the bacteria's. And when these guys go and infect a new bacteria, what they actually are doing is they're actually transferring DNA from the other bacteria to this new host. That's called transduction. Uh, it's a mistake. It's just an error. They just cut at the wrong place. There's generalized and specialized transduction. Obviously, you guys got to know this. A random piece of bacterial DNA is transferred during the lytic cycle. Specialized transduction occurs at the end of the lysogenic cycle when the prophage is excised and bacteriophage enters the lytic cycle. Does that make sense, guys? So, I think that's pretty good. Animal viruses. These guys use basically the same sort of pathway as those bacteriophages, but of course bacteria, it's kind of hard to leave them and get an envelope, and if they did, it would be of no use. The difference is those enveloped, that really helps them, and of course the fact that the animal cells are eukaryote and we don't have any cell walls. So, as far as the replication in an animal, we have direct penetration right there. Notice that the capsid lands there, and there is the nucleotide being inserted in, almost like the injection thing. Membrane fusion, and this is where we do get the capsid inside. The membrane fuses and brings the capsid in, which is immediately discarded by the virus. This one happens during like a phagocytosis or endocytosis, in which this is brought in, and again, the entire envelope, and it stays intact inside until the virus gets ready to leave. First he leaves his envelope, then he leaves his capsid. So the three mechanisms for animal is direct penetration, membrane fusion, and endocytosis. And as soon as they enter the cell and they get out of the capsid, they are called uncoated. Synthesis of DNA viruses of animals. Each type of virus has its own way of doing this. DNA viruses a lot of times will go to the nucleus, and because that's the place to be replicated in a eukaryote. RNA viruses will oftentimes do it in the cytoplasm, but there are exceptions. Smallpox is a DNA virus, and he does his thing in the cytoplasm. But in general, for rules of thumb, that's the way we'll consider it. Must consider mRNA synthesized and what serves as a template for nucleic acid. Yeah. Synthesis of RNA viruses. A lot of these are retroviruses, and this is wild because this kind of flouts one of the principles of biology, that the biologist hold dear. That principle says DNA makes RNA, which makes proteins. I won't write the whole thing out. But with a retrovirus, and HIV is a retrovirus, by the way, they use their RNA, and they have a special thing called reverse transcriptase. They take their RNA and turn it into DNA, and then they infect the cell. It's kind of an unusual approach, but apparently it works very well for it. So here's this guy. This is the budding off. You see the virus inside its capsid. It's already been assembled. It's going to the plasma membrane, and it's already put its spikes on there. So it's going to do exocytosis and take that bubble with it. And again, and there it's an envelope virion. And the DNA again is going to do in the nucleus. The RNAs are in the cytoplasm. How many viruses leave depends on the type of virus and the size of the cell that it's infecting and the health. Envelope viruses will oftentimes do this budding. Naked viruses can do exocytosis like that, or they can actually burst our cells. So that's possible as well. Persistent infections, there are some that we never get rid of. All of the herpes viruses we have forever. Varicella zoster, which is chickenpox virus, is a herpes virus. What happens to these guys? Do you guys remember the dorsal root ganglia? We've got nerve cells in there. So these guys infect those nerve cells. They cut open the DNA and then they insert themselves. And under the right conditions where the immune system is weak, they will take themselves out and come back and start an infection. That's where shingles comes from. It's varicella zoster reactivation. So these guys can persist, and all the herpes simplex 1 and 2 can do the same. Two primary, persistent or latent and chronic. Latent would be these guys here because they're laying low. Chronic is one that the immune system is not able to completely get rid of. I guess there was supposed to be a photo there. Chronic infections, they're recurrent or persistent over a long time. For whatever reason, the body can't do it. HIV would be a chronic infection. Well, we know why it can't do it, because the T helper cells are its target, and the T helper cell coordinates and activates the immune response. Some of these have no symptoms at all. Hepatitis C is the same way. It's a chronic infection. People get it and they may go many years without symptoms until it finally causes liver damage or liver cancers. Viral growth curve, inoculation. You have this many viruses. How come it goes down right there? Because you no longer have viruses. They uncoat, right? They're inside the cells. Eclipse, the virions penetrate into the cell. Burst, host cells release many, and the burst size of virions released per bacterium. And of course, these are all bacteriophage. Inoculation, eclipse, the entry, burst when the number of virions are big enough to burst the host cell. Burst size, however many that particular bacteria can hold. How do you grow viruses in the laboratory? We said that viruses aren't alive, they don't grow. You can't put them on a plate. The only thing you can do is infect. eukaryotic cells. I guess you could do back prokaryotes. Most of the time they're going to be eggs. Why eggs? Because eggs are the biggest cells in the world. Okay. Three types of media: mature organisms, cell cultures, or embryonated eggs. So if you look over here, this is a plate with E. coli on it. Some of these have the bacteriophage, and there's one called T4 that's grown on there. So this was not individual colonies of the E. coli. These are actually a bacterial lawn, and those holes are where the bacterial cells have been destroyed by the virus. They use different plants and animals to grow these viruses, and I use the term grow. I know they can't be grown. Culture, I guess, would be a better term. Laboratory animals cost a lot, and a lot of people don't like to see animals used like that. Culturing viruses in the chicken eggs, it's inexpensive, huge cells, no bacteria there. It's got stuff for the virus to, or it's got energy for the machinery. Fertilized chicken eggs are often used. They're the ideal site, and that's why when you go get your flu shot, a lot of times they're going to ask you if you have an allergy to eggs, because in order to make this vaccine, they have to culture the virus, and they do that in chicken eggs. And then they take those viruses and subject them to something that's going to actually break them into little pieces so that they're inactive. And then we insert those viral pieces into people, and that primes the immune system so you don't get the virus, in theory. Cells within chicken eggs, viruses replicate. Well, there's the egg yolk, there's the albumin, which is the embryonic chick, the amniotic fluid. So they put these viruses in different places. Viroids, virusoids, and prions. Viroids we've already talked about. Small circular pieces of, um, of RNA, and check out this effect. Look at those potatoes there, and look what happens when they have this viroid. May appear linear because of hydrogen bonding, and some of these guys can affect fungi, but as we said before, we won't go there. Prions are even wilder. What is more strange than an infectious piece of DNA? How about an infectious protein? You get a protein, and we remember proteins have different shapes. So let's say that's the normal shape for the protein. Here comes this prion. What he does is he makes this guy change shape. And it's just like dominoes. And that changes that so that it wouldn't work. Like, it'd be the same things if we got a metal file and filed off all the tips on our keys. You still got the key, but it's no good. It will not do what it was intended to do. Anyway. They call this prion. Oh, that's at the top. I'm sorry, we're actually talking about virusoids. Second type, pathogenic RNA. Subviral particles best described as non-self-replicating. Oh, wow. Do not concern yourself with that. That's the first time I've seen it. Let's get back with our proteins. Anyway, proteinaceous infectious agents. Yeah, it's an infectious protein. That is so wild. These guys So this is the PRP stands for prion protein. It is a normal protein that we have in the cells in our brain. Normally, it's going to be an alpha helix, but the prion protein, which is a disease form, the one that's folded wrong, is going to change it from alpha helix to beta pleated sheet. And by doing that, it's going to destroy its function. And that's the basis of mad cow disease and Scrapie and Kuru and a lot of other prion, Creutzfeldt-Jakob. So this is the problem with this stuff. What can you do about it? You can't do a thing about it inside the human body. We can destroy them with our sterilization technique, but you can't sterilize a patient. You wouldn't survive it. So prions are pretty bad. There's the cellular form with the alpha helices, and here's the prion form, which has totally changed. You can see those helices are now the beta pleated sheets. Spongiform encephalopathies. Bovine spongiform encephalopathy is mad cow disease. Variant Creutzfeldt-Jakob disease is a disease. Creutzfeldt-Jakob is actually the same disease, but it's inherited. Kuru, if you don't eat the brains of your enemy, you should not get Kuru. So if you have to kill your enemy, do not eat their brains. I'm serious. That's how that came about, and of course it's not really cannibalism. It's like ceremonial things in primitive societies. Anyway, these guys cause big holes to form in the brain. You get this from eating, transplant, contact with the mucous membranes. You can destroy these guys by incineration or autoclaving them in concentrated sodium hydroxide. Talk about pH of about 14. Yeah, that's fine. You can clean off things, but you cannot treat them. There is no treatment for this. The normal brain, and there's the one with Creutzfeldt-Jakob. Do you see the difference there? Normal brain, and look at these big lesions, those big holes in the brain. Creutzfeldt-Jakob disease itself is a disease caused by marrying your cousin too many times, and this is most prevalent with the Ashkenazi Jewish population in Eastern Europe. It's a problem. The variant Creutzfeldt-Jakob could be any disease, any prion disease that mimics it, only it's not an inherited thing that you get from your parents. I think that's pretty much everything right there, guys. This is kind of interesting. It's telling you how big the bacteria are and then comparing the viruses and the prions. And that's all right, guys. That's everything for exam 1. That finishes chapter 6. So we'll be seeing you.